Good morning, everyone. Thanks for joining us for Asthma Grand Rounds. Today, we're really privileged to have Dr. Lora Bankova, who's a member of the Allergy and Clinical Immunology Division at the Mass General Brigham. She's going to be talking about allergen and damage-sensing epithelial cells in the airways. Lora is an assistant professor in the Division of Allergy and Immunology in the Department of Medicine. She's done really groundbreaking work understanding the similarities and differences in how the airways and the sensory system cells detect allergens and damage. Her work actually established links between epithelial cell dysfunction in the nose and conditions associated with smell and taste dysfunction, such as persistent COVID-19 related loss and chronic rhinosinusitis. She's published widely, including in major journals such as Science Immunology. She's a PI of an NIH R01 and an R21, and we're really privileged to have her talking about her groundbreaking work today. Just a few housekeeping things before we have the privilege of hearing Lora. So this is for the live webcasting audience. You can submit a question to our speaker by entering it into the chat. We'll go through those questions at the end of the session. CME credit is being awarded by Mass General Brigham Office of Continuing Professional Development. That's MGB, Mass General Brigham, CPD for Continuing Professional Development at mgb.org. And these are the instructions to set up texting for attendance. And I will put the phone numbers in the chat. The major thing you want to know is that if you haven't done this before, that you want to send a code for attendance to 857-214-2277. As I said, I'm going to try to put that into the chat so everybody can access that number if you didn't get a chance to write it down yet. And the learning objectives here: identify allergen damage sensing epithelial cells in the airways, be able to describe the role of allergen damage sensing epithelial cells in rhinitis and asthma. These are my conflicts, and Dr. Bankova doesn't report any conflicts at all. If you have any technical problems, you can email BWH webcast troubleshooting@partners.org. And to remind you, if you have any problems with this, you can write to MGB-CPD. Remember, at Mass General Brigham, Continuing Professional Development at mgb.org. And so I'm going to turn the mic over, as it were, to Dr. Bankova. Thank you again, Dr. Bankova, for joining us.
Good morning, and thank you really for the invitation to present here. So Asthma Grand Rounds is how I grew up through the fellowship here, and it was always the highlight of my education, both as a fellow and then moving forward. I always looked up to all the amazing speakers at Asthma Grand Rounds. Okay, so so I'm going to talk about epithelial cells and a particular subset of epithelial cells called tuft cells today. But before I get into that, I was just going to introduce the airway epithelium as really the forefront of sensing and initiation of any immune response really in the airways. The epithelium was initially considered a really simple barrier between the inhaled air and the submucosal immune system. As you can see here, it's recognized that the epithelium is important because there's all these cytokines coming from epithelial cells, TSLP, IL-25, and IL-33 that I'm sure you're all very well aware of as initiators of innate immune responses. But it was also always drawn in these nice reviews in Nature Reviews Immunology and other established journals as a very simple epithelial barrier of ciliated cells and secretory or goblet cells. But with the advent of single-cell sequencing and more advanced imaging technologies, we started to understand that there's a huge diversity of epithelial cells, not just of immune cells. And they are driven by unique receptor expression and also specialized mediator profile. So fast forward only 4 years between that previous slide and this slide. So in 2019, Nora Barrett, who is a member of the Asthma Center and was my KOA mentor, published this review in the Journal of Allergy of Clinical Immunology, where you can see that the diversity of the airway epithelium has jumped forward quite a bit. And there are distinct epithelial cells here. So in addition to basal stem cells, there's ionocytes that were discovered in 2018, neuroendocrine cells that generate neuromediators, and then the goblet cells, and the cell that I'm going to talk about, which is the called brush, tuft, or solitary chemosensory cell, depending on which paper you read. Until recently, when they really became the tuft cells, and that's how they're referred. So here's a tuft cell that has a unique structure. And what did we know about tuft cells before a few years ago when the field really exploded? It was known that they're a rare epithelial cell type, so less than 4% of epithelial cells. They have common features that they share throughout the whole body. They were initially described as bitter tasting cells, but many of them don't actually express bitter taste receptors. They do all, however, express that taste signaling cascade throughout the body. They are dependent on a transcription factor called POU2F3 for their development. So if you see mice that lack POU2F3, they lack tuft cells, and they have these unique microvilli over here. So it's a tuft of brushes on the surface of these epithelial cells, and they are distinguished just by their morphology from other epithelial cells. And then here on the right, what was known about tuft cells mostly in other compartments of the body, not just the airway epithelium, was that they detect bacteria and endoparasites. They generate a very unique combination of mediators. So the motor neuron neuromediator acetylcholine, leukotrienes and prostaglandins that we previously knew mostly as mediators generated by leukocytes, and the epithelial cytokine IL-25. Through these mediators, tuft cells can activate immune cells, mostly through IL-25 and cistern leukotrienes. Through acetylcholine, they can directly send signals to neighboring epithelial cells to promote mucociliary clearance. And they have been shown to also activate sensory nerves, which has mostly been linked to acetylcholine. But how tuft cells really came into the center stage of mucosal immunology was in 2016, when there were 3 high-profile papers that defined these previously enigmatic chemosensory cells in the gut as the dominant source of IL-25, which is shown here in white over here and in red in the picture below. And we collaborated with a group of Jakob von Moltke, who had published one of those papers. And together with Nora Barrett, we showed that the tracheal epithelial chemosensory cells that were previously called cholinergic brush cells are, and shown here cholinergic is because they express CHAT, which is choline acetyltransferase, the enzyme required to generate acetylcholine, are also the major source of IL-25 in the airways. So this is again shown in the trachea, and IL-25 marks in a red fluorescent protein expressing mouse marks the nucleus in this picture. So, so we found that these cells are IL-25 expressing, acetylcholine expressing in the trachea, but we really wanted to know how they're engaged in the response to allergens, because in the gut, this was mostly response to endoparasites. So tuft cells, again, going back to another review, are situated on the airway epithelial surface. And here's a house dust mite, one of the many particulate matters that we can inhale. And we wanted to know if an allergen can activate the tuft cells. And we really have been trying to get at that from many different perspectives. And I will jump to some of our most recent data and then go back to our initial data showing this connection, just because it is so obvious and so beautiful to highlight it using this system, where a very talented postdoc who joined my lab a few years ago, Alex Pernes, decided to use the school system where you can use a calcium sensing protein, GCAM. So this is a protein that reacts with the change in fluorescence over here, when there's an increase, intracellular increase in concentration of calcium. So you would have a situation where you can take, what he's doing is he takes a mouse trachea or a mouse nose, puts it under a microscope, and he can add different ligands to the solution under the microscope. And if there's a change in flora, if there's an activation of the cell, so if there's a flow of calcium within the cells, he would see a fluorescent signal. So the cell would that is reacting would turn on fluorescence and would look white. So what Alex did is using this system, he first added ATP, the prototypical danger signal. And you can see here how the tough cells, these are only tough cells because they're marked by the cholinacetyltransferase recombinase, are lighting up within 30 seconds. He then added Alternaria in the same system. And here, initially, nothing was happening. But if you keep looking at 150, 190 seconds, the tough cells start reacting. And he did the same thing with house dust mite allergen. And here we have a slightly different response where the cells are getting activated at 90 seconds. But again, you can appreciate that with ATP, there's a really fast response, while with Alternaria and house dust mite, there's a delayed response, suggesting that there's different pathways that might be activating these tough cells in the whole tissue. So he has done measurements of really the fluorescence intensity and the time, which is on the y-axis here, and the time to activation. And you can see that there's this really fast response to ATP. There might be a second wave of activation. With Alternaria, there's much more of a slower but sustained activation of cells over time. And with house dust mite allergen, there's an early peak and later activation, suggesting that these all might be triggered through different receptors, different receptor pathways. But interestingly, so many different ligands can all converge into the activation of these tough cells in the airway. So here we can see that he's done this with ATP, with succinate, in the trachea, which was actually found by Alex himself in his previous lab as a really specific and strong inducer of tough cell responses in the trachea. He can do the same thing with denatonium, which is a bitter tasting ligand, and then Alternaria, house dust mite, and also with papain and a part 2 agonist. And these are measured responses here to all of these allergens. And also what we have been doing is measuring the time until reaction. So the thing that you saw in the previous video, where there's really different waves of activation of the tough cells in the airways, depending on the specific type of ligand. And we'll be working on trying to figure out what these receptors, these specific receptors might be that induce the response. And then something else that Alex has started doing and is really cool is trying to understand if allergens can induce a spread of signals that starts from the tough cell and spreads to neighboring epithelial cells. So the way he does that is he takes, again, a cholinacetyltransferase mouse. So in this mouse, only tough cells in the epithelium, because in the epithelium, tough cells are the only cells that express cholinacetyltransferase, and crossed it to a tomato mouse. So in this mouse, the tough cells would be red. And then he loaded the whole tissue with a calcium dye, so you can see the activation of not only tough cells, but neighboring epithelial cells as well. So doing that, when he applies papain, focusing on the tough cells, you can see that there's these cute waves that start from the tough cells or spread of signal and spread across the whole epithelium. And he has done this also with house dust mite. And you can see tiny waves here, again, starting from some of the tough cells. And also with Alternaria, this is a more zoomed out view. And again, if you focus on the red tough cells, you can see the spread of signal that starts from tough cells and goes to neighboring epithelial cells. So really suggesting that tuft cells are not only sensors for allergens, but they also serve as hubs for spread of the signal across the epithelium. So there might be other cells in the surrounding space, in the surrounding milieu, that are getting activated. But if the activation signal goes to the tuft cells, then they can provide a secondary spread across the epithelial surface. But thinking again about the function of tuft cells and how this all happens, I started this off by this presentation by saying that tuft cells are rare epithelial cells, and they account for less than 4% of epithelial cells in most mucosal compartments. So when Nora and I started studying this compartment, studying tuft cells, one of the things that we realized early on is that tuft cells are really absent in the lung. There's none of them in both mice and humans. They're also absent, so they're absent in the alveolar space, they're absent in the small bronchi, they're extremely rare in the larger bronchi. They're rare but present in the trachea, so these calcium signaling experiments that I showed you, many of them are done in the trachea by Alex, but what we realized is that they're much more abundant in the nose. And we frequently ignore the nose unless we are folks like Tanya Laidlaw and a lot of our ENT physicians who are interested in the nose as a separate compartment where you have a lot of type 2 inflammation. But in general, in mucosal immunology, when you say airway responses, people mostly focus on the lung. So when I started looking for tuft cells and trying to understand how tuft cells really respond to environmental signals and allergens, I was sort of shocked to notice that there's this big difference between the percent of tuft cells in the trachea. So this is, again, using this choline acetyltransferase reporter mouse. And within the epithelial cell compartment in the trachea, there's 0.5% of epithelial cells are tuft cells. But in the nose, you have 7%. And sorry, this is not among epithelial cells. This is among non-immune cells. So in the nose, 7% of non-immune cells are what looks like tuft cells. But in the nose, there's two different populations of tuft cells. So there's the ones, if you want to call a tuft cell just based off of its morphology, there's the classical looking tuft cells, which have high forward and side scatter by FACS. And then there's this population of really tiny lymphocyte gate-looking cells that mostly come from the olfactory mucosa in the nose. And I have several slides to introduce the olfactory mucosa to you later, but these two appeared like they're both cholinergic, but one of them maybe was not quite what a tuft cell should be. So we sorted both of these and submitted them for bulk sequencing. That was a few years back when people weren't doing single cell sequencing for everything. And what we found was that the tracheal, nasal, respiratory, nasal, olfactory CHAD positive cells, we weren't calling them all tuft cells at the time, really clustered together if you took the 100 most variable genes. And what we were really surprised about was that among the genes that really defined what makes a tuft cell were three members of the cystine leukotriene generating cascade. ALOX5AP encodes FLAP. ALOX5 encodes 5-LO, which is inhibited by xylulin for the asthmologists. And LTC4 synthase is the terminal enzyme that directs the generation of LTC4. So really, here's an epithelial cell that is defined by its expression of leukotriene generating enzymes. So we thought that was interesting, and it was something that we can work with from two different perspectives. So one was that it provided us with a population of tuft cells that is sufficiently abundant to develop an ex vivo or in vitro system for stimulation where we could add allergens or other ligands and look for a readout, which is a mediator readout. So here in this case, the cystine leukotrienes. So we did exactly that. We took the mouse nose And we sorted the chat positive cells, which are tough cells, and we would get about 20,000 cells from one mouse, which for tough cells is an amazing number of cells. And we were able to put them in a 96-well plate and stimulate with ligands and look for activation. And when we did that, we started off with ATP because several ATP receptors are highly expressed in tough cells, and we really wanted to define the system. And with ATP, we could get about 10 to 20,000 picograms per ml per million cells of cystine leukotrienes, which is really similar to what you would get with eosinophils and with mast cells. Oops. And then we compared this generation of cystine leukotrienes because this was measured by ELISA to that in leukotriene C4 synthase deficient mice, just to confirm that we're really measuring leukotrienes, and also P2Y2 receptor deficient mice, as we had hypothesized that this might be the receptor that drives the activation of tough cells in response to ATP. We confirmed that, and then we wanted to know, really, do allergens really activate tough cells directly in this in vitro system where we only have tough cells and no other epithelial cells? And when we did that with house dust mites or dermatophagoides pteroniecinus and with alternaria, we found a nice dose-dependent increase of cystine leukotriene generation. And if we pre-treated the cells with a P2Y2 receptor inhibitor before we gave them alternaria, so alternaria here in the blue bar, and pre-treatment in the gray, pre-treatment with the P2Y2 receptor inhibitor in the gray bar, we found that there's a reduction in the generation of cystine leukotrienes by tough cells, which really goes back a little bit to what I showed you in Alex's new data. What we think is that there's really a receptor that on the surface of tough cells that is activated by allergens like alternaria and dermatophagoides pteroniecinus, and activation of the tough cells leads to translocation of ATP outside the cell and activation of the P2Y2 receptor, which then induces a feed-forward loop of activation, generation of LTC4, which then gets exported outside the cell and can cause downstream effects. So what are these downstream effects that are driven by tough cells? So Nora Bankova had shown in some of her earliest work that cystine leukotrienes can activate dendritic cells for Th2 priming. LTC4 and IL-33 cooperate to activate ILCs in the airways, and then LTC4 and IL-25 can also cooperate to activate intestinal tough cells. So we wanted to know what will happen if we look at LTC4 and IL-25 together and what their effects are going to be in the tissue. Specifically not in the gut, where this was shown by Jakob, but what happens in the airways, where tough cells are directly activated by allergens. So to do that, we gave mice either only LTC4, so the first leukotriene, the biosynthetic product of the pathway, or IL-25. And everybody who's done any sort of reading into the classical papers of cystine leukotrienes and what they do to human or mouse airways knows that LTC4 on its own does not really induce inflammation in a naive lung, in a lung where there's no pre-existing inflammation. And IL-25 is also known and as a pretty, among the epithelial cytokines, is probably the one that has the least pronounced effect when inhaled or when people are exposed to it alone. So we found the same thing if we did only LTC4 or only IL-25, looking here in the lungs, here the airway spaces, and the bronchoalveolar compartment where most of the immune cells would come in. So you can see that with LTC4, nothing, there isn't very much inflammation. Really, maybe there is a little bit of remodeling in some submucosal immune cells with IL-25. But if we gave LTC4 and IL-25 together, there was this dramatic increase in inflammation with only 3 doses. And you can see the same thing when you look at the mucous cell metaplasia, so goblet cell development with porotic acid shift. There's a huge increase in the number of goblet cells in the airways. And when we did facts to understand what kind of cells are recruited to the tissue, and also multiplex ELISA to determine what kind of cytokines are induced. We really found a 60-fold increase in eosinophils, a dramatic increase in proliferation of innate lymphoid cells, and pretty significant induction of IL-4, IL-5, and IL-13. And what I'm not showing here is that when we did the multiplex ELISA, we found no induction of any other cytokine. So there is really a very strong type 2 polarization of the immune response. We didn't get any IL-17, no interferon gamma. This was mostly type 2 inflammation. So what we showed really is that the two mediators of tough cells, LTC4 and IL-25, when they get released, they induce a really strong response in the airways. But what happens if we do it the other way? Because this is just a pharmacological dumping of mediators down the airways system, which doesn't always work this way in the whole tissue. So what we did is we used a mouse that Dr. Boyce had generated that can that has LTC4 synthase flux. So we can delete LTC4 synthase, the enzyme that drives LTC4 generation, specifically in the tough cells. So we did that using our favorite chat-cree system. So again, driving with cholinacetyltransferase. And we compared wild-type mice to mice that lack tough cells to these new chat-cree LTC4 synthase flux mice, where only tough cells do not generate cystoleucotrienes. And when we did that, whether we gave mice, if we gave mice one dose of an allergen, Alternaria, and we looked 1 day later in the bronchoalveolar lavage, or if we did 4 challenges with Alternaria and looked 9 days later, with both systems, we found that there was a reduction in eosinophil recruitment to the lung. So here first only in the early phase, but also in the adaptive phase of allergic inflammation, there was a reduction of eosinophils coming into the tissue in mice that lack tough cells or mice where tough cells cannot generate cystoleucotrienes, suggesting that really tough cell-derived cystoleucotrienes are required for contribute to the development of allergen-induced lung eosinophilia. So tough cells both sense the allergen and their mediators drive the inflammation downstream. But what happens if we if we inhibit IL-25? So I showed you that LTC4 and IL-25 really cooperate. So would we get complete reversal of the inflammation if we inhibit both? So I'm jumping over one slide here because there's nothing to show there. We looked at that in the lung, and in the lung, we didn't find anything beyond what we were finding with LTC4 synthase deletion if we also inhibited IL-25. But when we looked in the lung draining lymph node, so here, if we gave mice a dose of IL-25 inhibiting antibody before treating with Alternaria, and then again a day later, and then if we looked in the lung draining lymph node 24 hours later. I'm going to walk you through these guys here because it's a little confusing. So this is in white is wild-type mice that were given PBS or Alternaria and the IL-25 blocking antibody. There's a little bit of a reduction in immune cells in the lymph node. And then we're looking at the tough cell C-cell T deficient mice without IL-25 blocking antibody and with IL-25 blocking antibody here. You can see that there's quite a significant reduction in the lymph node response to allergen, the lung draining lymph node response to allergen, if you both deleted tough cell C-cell Ts and inhibited with an anti-IL-25 blocking antibody. And this was particularly pronounced if you look at the CD4 T cells coming into the tissue. So really tough cell-derived cystoleucotrienes and IL-25 together drive type 2 inflammation in the airways is what these studies showed. But the question still remains, how does this work? Because in my previous slide, I showed you that the tough cells in the trachea are really rare and they're practically absent in the lung proper of the both bronchial space and alveolar space. So there must be some link where tuft cells are sensing allergens in the nose or in the trachea, and they're sending links to the lungs. So initially, to answer that, we started looking more carefully at the nose. And this is, again, work from Alex, who is an amazing microscopist. So he did this cool system where he took the mouse nose, split it in half, so you can put half of the nose under a microscope, and he stained for sensory neurons and for tuft cells and for with beta tubulin for respiratory epithelial cilia. So one thing to remember about the mouse nose, unlike the human nose, is that the mouse nose has 50% olfactory epithelium, while we as humans only have about 10% of olfactory epithelium in the nasal mucosa. So the touch one here stains not only sensory neurons, but also olfactory neurons. So this part in the back, sort of the northwest corner, is the mouse olfactory epithelium, and the southeast is the respiratory epithelium. So he did that in a chat GFP mouse, so where chat would mark the tuft cells. And you probably can't appreciate all the tuft cells in this merged picture. So here, looking at the same picture, but now taking away the olfactory staining and respiratory staining, you can appreciate how abundant the tuft cells are. So in the respiratory epithelium, they're scattered, similar to how rare and scattered they are in the tracheal epithelium, but in the olfactory epithelium, they're really, really abundant. And again, I showed you this picture earlier. They have different morphology in the two compartments. But one of the things that we were really excited to find in collaboration with Tanya and Katie Buckheit, and also our wonderful ENT colleagues who have provided samples from surgeries, we found that in human olfactory epithelium, so here NCAM is a marker for neurons, including olfactory neurons, and OMP is olfactory major protein, which marks mature olfactory neurons, here the yellow guys, and looked in this human olfactory epithelial sample for tuft cells staining with that villain. We can see, similar to the mouse, scattered tiny, similarly shaped tuft cells across the olfactory epithelium in humans. And I have this one picture of a respiratory human tuft cell, and I have to say that we stained about 20 or 30 slides to find these two good-looking respiratory tuft cells, while in the human olfactory epithelium, almost every tissue slide that we looked at had an abundant number of tuft cells. So tuft cells are abundant not only in mice, in the olfactory portion of the nose, but also in humans. So we were interested to find out more about these olfactory tuft cells and how they contribute to the whole mucosal response, like the whole airway response to allergens. And again, we frequently think about the nose as just part of the conducting airways, but I would like to convince you today that it's not only a part of the conducting airways, but it has a lot of other functions that are quite unique and difficult to put together in one system, and really interesting. So when you dissect the nose coming from the viewpoint of mucosal immunologists, we're really surprised to find that there's this whole other compartment of the olfactory epithelium, which again in humans is possibly only 2 to 10%, but might cover a larger area. It's a little hard to tell what is olfactory epithelium and what is not, because macroscopically it looks the same. But the olfactory neuroepithelium is really a unique sensory and epithelial organ. So the olfactory sensory neurons are the only actively regenerating neuronal population, and they're located here at the very top of the nasal mucosa, embedded in a space where they're surrounded on the apical surface by epithelial cells. And these epithelial cells are what I'm going to talk more about in the following slides, but some of them include the tuft cells here on that surface. So importantly, the olfactory sensory neurons send projections, they're true neurons. So they're embedded in this epithelial layer. They extend their projections into the luminal space where they can detect odors. And then they send their axons through the cribriform plate to the brain where they make connections in the olfactory bulb to the central nervous system. The other thing that's quite unique about the nose is that on top of having this olfactory sensory system, it also has a second sensory system, which is the peripheral sensory system that comes from the trigeminal nerve. And the trigeminal nerve has three components, as you all know, and some of them innervate the nasal respiratory epithelium, but many of them also extend into the olfactory space. And then just to introduce the olfactory neuroepithelium and its more complex structure to you. So again, I'm sure that many of you are very well aware of how the respiratory epithelium is organized in the lower airways. It's very similar in the nasal mucosa. So there's stem cells that give rise to ciliated cells, secretory cells. There are a lot of goblet cells in the nose at homeostasis because your nose needs to get rid of particulate matter at homeostasis. There are ionocytes and tuft cells, and they're all derived from a population of respiratory stem cells that are keratin 5 positive. In the olfactory epithelium, there's also a population of keratin 5 positive stem cells that are very similar to the respiratory stem cells transcriptionally, but they're quiescent in the olfactory epithelium. And there's a different population of globose basal cells that are constantly proliferating, and they give rise to the olfactory sensory neurons, which are shown here in different stages of differentiation from immature sensory neurons to mature sensory neurons, which are shown in purple. The olfactory neurons are pretty much the only population of neurons in your body that is constantly being regenerated. So they have a half-life of about 90 days, and then they're constantly dying off, being repopulated with new olfactory neurons that send their axons. These newly derived olfactory neurons send their axons to the brain, and this is a system that works amazingly well. So the stem cells in the olfactory epithelium also give rise to sustentacular cells, which are the epithelial cells that provide the structure of the olfactory epithelium. And they're the ones that get infected by the SARS-CoV-2 virus. And there are also several other populations of epithelial cells that are well differentiated and also similar to the respiratory specialized epithelial cells. So there's the tuft cells and the ionocytes. The ionocytes in the olfactory epithelium have a slightly different structure, but transcriptionally, again are almost identical to the respiratory ionocytes. And there are also ductal cells that are line the ducts of the Bowman's glands, which are the secretory glands in the olfactory epithelium. So a huge diversity of epithelial cells that largely has been ignored by the mucosal immunologists because we don't think about the olfactory epithelium, and by the olfactory biologists because they don't think about epithelial cells. So in a wonderful collaboration with Adam Haber, we looked by single cell sequencing at what happens in the whole nose if we gave mice an allergen, and we did 1 dose of Alternaria or 2 doses of Alternaria. And with single cell sequencing, we were able to capture all epithelial cells and immune cell subsets. So blue is epithelial, purple is olfactory neurons, and then all the other colors are the different types of immune cells. And with this data, we were able to assess the changes in proportions of the different types of immune epithelial and olfactory neuron cells. And as we expected, so this is Adam doing a fancy calculation where you can see the change in the log-fold change in the abundance of different cell types compared to naive. So the further to the right, the bigger increase in that particular population. So we found that, as expected, there was an increase in glandular mucus cells and in goblet cells. But what we were more surprised to see here is that there's a big increase of basal cells, and that was more pronounced than the increase in any of the immune cell populations. And we really don't see that much of a, we know that basal stem cells increase in number, but such a dramatic increase in basal stem cells that is roughly almost the same as what we see as goblet cell metaplasia in the nose was quite surprising to us. So we then used flow cytometry to do this in a cheaper and scaled up way, and to look for stem cell numbers focused on proliferation specifically. So by flow cytometry here, what we did is we looked in the epithelial cell compartment for keratin 5 positive cells that would also be proliferating with Ki-67. And we found that Alternaria really induces a strong proliferation response. So there's about a doubling of the stem cells after a single inhalation of Alternaria. And to our delight, this was reduced in tuft cell deficient mice. But surprisingly, we thought this was going to be like tuft cells talking to ILCs and feed forward loop with IL-13 receptor driven proliferation of stem cells. We found that IL-7 receptor deficient mice, where there's no lymphocytes, and specifically no innate lymphocytes, and STAT6 deficient mice really had no reduction of the stem cell proliferation response, suggesting that there's like this strong allergen-induced nasal stem cell proliferation that depends on tuft cells, but not on ILCs or on STAT6. So then we wanted to see how does that differ in olfactory versus respiratory epithelium. And the top here, where olfactory marker protein marks the olfactory neurons, is the olfactory epithelium, and then the bottom is the respiratory epithelium. So we decided to look at these two compartments separately and do this with both Alternaria, but also another damage signal, though this time ATP, because we knew that this also activates tuft cells. And what we found here, surprisingly, was that there was a much stronger response in the olfactory epithelium than respiratory epithelium. So while there was a 20 to 30-fold induction of proliferation in the olfactory epithelium, there was much weaker response in the respiratory. So there's definitely proliferation in both compartments, but much more pronounced in the olfactory epithelium. So we know that this was tuft cell dependent, and we know that it doesn't depend on type 2 inflammatory loops in the innate phase. So we wanted to know, well, is it still dependent on cystine leukotrienes, because they're our tuft cell mediator. And to our delight, it was. So here, this is shown by histology and by FACS. Histology on the left, FACS on the right. So if you look by histology, there's an increase in these green cells, which are the Ki-67 positive stem cells. And they're marked with the arrows here, so it's more obvious. And this is on the top in wild-type mice, and on the bottom in the tuft cell C-cell-T deleted mice, there's really no induction of this proliferation response in the basal stem cell compartment. And we evaluate that also by FACS, again, to scale up our evaluations. And we found that both with ATP and with Alternaria, there was a reduction in olfactory proliferation in response to Alternaria in these tuft cell C-cell-T deficient mice. And if we look in the respiratory epithelium, interestingly, we don't find that HP induces a very strong proliferation response here, suggesting that there's different pathways that trigger this stem cell proliferation response, but there was a reduction in respiratory stem cell proliferation with Alternaria in the tuft cell deficient mice as well. So obviously we wanted to understand how this works, because it's not driven by immune cells, but it is still C-cell-T deficient, C-cell-T driven and tuft cell driven. And one of the big problems that we came across here is that tuft cells and stem cells in the olfactory mucosa are really far apart. So here are our tuft cells that are marked with a villain. at the very apical surface where they can sense allergens, they can produce their cystine leukotrienes, but then there's multiple layers of epithelial cells and olfactory sensory neurons that separate them from the stem cell layer. So how are these stuffed cells activating the basal stem cells? So we go back to some cool things that Alex has done again, now with the collaboration with Isaac Chu's lab. Isaac had generated a CCL2 Cree mouse that we crossed to tomato. So this way, we can look for some of the receptors that are expressed that would be activated by the LTC4 generated by tuft cells, and trying to understand which receptor is going to be responsible for the activation, and trying to understand like where the cells that might be driving this response. And I'm jumping straight to CCL2 receptor just because for time's sake, but we had looked at each different receptor and found that none of the other receptors are responsible for this response, for this like driving the response to cystine leukotrienes. And with this mouse, what Alex found is that there are these finger-like projections that go from the basal stem cell layer to the surface layer where the tuft cells would be. And these represent sensory neurons, which I'm going to be convincing you in the next few slides. And then what Alex then did is he crossed this tomato mouse where CCL2 expressing neurons, or all cells, would be marked red, to a mouse where tuft cells would be marked green. So CHAD GFP mouse. And here you can see that the tuft cells are at the apical surface as expected. They're the sensory neurons here that cross this the whole layer, the whole olfactory layer, and then there's the stem cells at the basal layer. And what is really pretty and like convincing here is that these CCL2 expressing sensory neurons are really sending projections to many of the tuft cells at the apical layer. They have tight connections to some of them, and then they cross the whole olfactory sensory neuronal layer and then wrap around the stem cells. So they really wrap around the whole stem cell layer at the basement membrane and provide a link from the allergen sensing tuft cells to the stem cells in the olfactory stem cell compartment. So then the next question is, if these CCL2 receptor nerve fibers bridge the distance from stem cells to tuft cells, where do they really come from? So to answer that question, Alex gave wheat germ agglutinin, which was fluorescently labeled, to the nose of mice, and then looked in the trigeminal ganglion where we expected them to be coming from, and confirmed that really this compartment is innervated by trigeminal nerves that come into the tissue. So then we wanted to know if really these are sensory neurons that are CCL2 positive. And to do that, Alex looked in together with a summer student who was really invested, Eric Zhu. We looked at sensory neurons that would be marked as NaV1.8, which is a nociceptor pain-sensitive fiber marker, marked here in red in the trigeminal ganglion. And he looked for co-expression of this marker with CCL2 receptor and found that there's a really nice overlap where a large proportion of the CCL2 receptor positive cells are also positive for this pain nociceptive fiber marker. And he confirmed that these are C fibers in the trigeminal ganglion that really are positive for the CCL2 receptor, highlighting a really unique expression of a receptor that we previously have thought of as really coming in from endothelial compartment and a lot of immune cells. Now here in the olfactory and nasal epithelium, this is also a receptor that's largely expressed by pain-sensitive nociceptor neurons, and this lines up with work that Isaac had done showing that together with Dr. Austin, actually, where they showed that this receptor is responsible for itch in the skin, but it was not clear what this receptor might be doing in the airways. So to understand that, we first took mice that are deleted for all nociceptor neurons. So these are NaV1.8 Cre-DTA mice. So they have no nociceptor neurons coming into the olfactory epithelium or the respiratory epithelium. And when we looked for the olfactory stem cell proliferation, we found that there's a reduction of both ATP and alternaria-driven proliferation here. And this was true in the olfactory epithelium, but surprisingly, really not true for the respiratory epithelium. So the sensory neurons direct stem cell proliferation in the olfactory, but not respiratory epithelium. So then we cross these mice, so NaV1.8 mice, to the CSOT2 receptor expressing flox mice. So here we only delete CSOT2 receptor in the sensory neurons. And we found the exact same thing here. So olfactory stem cell proliferation is reduced in mice where CSOT2 receptor is deleted in sensory neurons, but only in the olfactory epithelium, not in the respiratory epithelium. Finding this really unique new role for CSOT2 receptor in regulating airway responses to allergens. And I will quickly show you this data just to confirm that this is driven by sensory neurons. Alex also looked for neuromediators that can be responsible for that and found that CGRP really is the driver of this response here. And just to end that with some prettiness, Alex wanted to really confirm that what the innervation of the nose is. Because one of the things that we are still struggling to understand is how is it that there's this difference in olfactory versus respiratory epithelium. Why would the sensory neurons be controlling the proliferation in the olfactory, but not in the respiratory epithelium? The stem cells are the same. Is it possible that the innervation is different? And it really isn't. So here's the trigeminal ganglion. This is a whole mouse head that was cleared, and he did whole nasal mount looking for sensory neurons in a mouse where the sensory neurons are marked red. And you can see that they extend both to the olfactory and respiratory epithelium. And if you look at this image that zooms in on this compartment, you can see that there's sensory neurons that go to the olfactory epithelium, which is at the back of the mouse nose, really infiltrate into the epithelial space there. But if you track these neurons further, they also go to the respiratory compartment. So the innervation doesn't appear so far to be all that different. And we're going to have to understand why this response is not the same. So what I'm hoping to have shown you here is that really allergens activate tuft cells for a feed-forward loop of activation to release ATP and generate cysteine-leukotrienes that then in the olfactory epithelium specifically activate the CSLT2 receptor on sensory nerves. And then the CSLT2 receptor triggers the release of neuromediators that drive the proliferation of the stem cells. And how is that relevant to human biology? One of the things that my ENT colleagues tell me is that a lot of the polyps come from the upper compartment, from the superior compartment of the nose, where the olfactory epithelium is really concentrated in humans. And Nora Barrett and Dan Dwyer have shown that one of the prominent features of rhinitis is stem cell proliferation. And again, tuft cells are really abundant in that compartment. So it's possible that this is what's driving some of the polyp formation in humans. And then how does that relate to asthma? It's a little hard to find research on specifically how to answer that question. But if you go to the American Lung Association, currently they tell you be careful with holiday scents and lung disease as lung disease triggers. And I found that like some of these papers are so amazing from like the 1980s. So this is FEV change in somebody who was put in a tent and given perfumes to smell. And there's a drop in FEV1, and that was done in multiple patients. And if you, on first exposure, there's a huge first and second wave response. Then they were treated with saline to see if they can reduce the response with cromolyn, atropine, and metoprolol, and none of those treatments reduced the FEV drop. The only thing, I didn't highlight that. The only thing that dropped the response, that inhibited the response, was closing the nose during the challenge, which really suggests that the olfactory epithelium has more functions, more effects on asthma than we really appreciate, but they're very difficult to study. And this is the percent of reactors in this study who reacted to different scents. So really suggesting that, and I think we know that from practice, a lot of asthmatics get triggered by perfumes. That's why we're not supposed to wear perfumes in our clinics. And the mechanism we have possibly started to scratch the surface on. And with that, I just want to acknowledge Jeff and Penny Vinnick Center, everybody in my community who has contributed to supporting me and the research done here, especially Alex, Southnut, Caitlin, and Dante, and Eric, who joined in the summer, our ENT colleagues and my collaborators, both at the School of Public Health and Harvard Medical School. Thank you.
Lora, thank you so much. That was really an outstanding presentation, and the way you followed this all through is really just a testament to kind of the work, the outstanding work you're doing. I was gonna, some questions are starting to come into the chat. And one of the questions was, have you tested any non-allergenic stimulation of tough cells like ozone, cigarette smoke, etc.?
We have not tested ozone. So, right, so we've tested ATP, UTP, so non-allergenic damage, some of the bitter tasting substances, succinate. We have not tested odors. I don't exactly know how we would test for them, but Alex is he's very inventive. So I'll talk to him about that, because some part of the problems with the system is that we have to do that in a system where we add the ligands in a water bath. So it's a little difficult to do that. But we can definitely try to do like look for some of these responses, like to cigarette smoke and ozone in a chamber, looking at the whole mouse. What I was more thinking of, like how the tough cells might affect the response to odors and odorants, is through this remodeling response, where remodeling of the whole olfactory epithelium might lead to the generation of new olfactory neurons that have different sensitivity compared to what you normally get with the normal regeneration response in the olfactory mucosa. So again, olfactory neurons get repopulated every 90 days. And this is a very precise process where they have to send their axons to the right place in the olfactory mucosa. So if you have new olfactory neurons that are a little confused because of the allergen or damage triggered remodeling in the nose, you're going to have a much different, you might have a much different response to non-dangerous odors.
Right. A question I had. So, considering everything you've shown about these LTC2 receptors, while montelukast blocks cis-LT1 receptors, the 5-lipoxygenase inhibitors, xylutrin blocks leukotriene production. Has that been shown to change our ability to sense through these mechanisms?
So I think you have shown some of these studies where 5-xylutrin is a much more potent medication when it comes to, like, well, not just asthma, but also for polyps. And then also Eric Van Dalen has actually done a specific study on what happens if you do xylutrin in patients with AERD to their sense of smell. So does it only affect their asthma or their asthma and their sense of smell? And the answer is yes, it improves their sense of smell much more so than montelukast. I
would have thought that it might reduce their sense of smell if you couldn't stimulate these receptors on the on these neurons, right?
Uh, Right, but if this is an aberrant response where
Hmm.
you're aberrantly, so stimulating the sensory neurons leads to an aberrant response to the allergen inhalation, then you
Hmm.
prevent the these aberrant response, and then you should have a better sense of smell. I think what I would like to do is to see what happens if you treat people with xyluten to their sensitivity to odors,
Yes.
to perfumes. And I looked for any data like that, and that has not been done.
Okay. All right. Well, we reached the top of the hour. This was a fantastic um, review. Thank you so much, Laura. And to
Thank you.
everyone else, again, I put in the chat if you want to get CME credit to the phone number to dial, sending that code, and you'll get a certificate from the CPD organization at MGB. Thank you all again.
Thank you.
I'm looking forward to seeing you at the next Asthma Grand Rounds. Take care and have a good weekend. And Laura, thank you again.
Thank you.